Hola, Team Bio. Okay, so far we have talked about the history of kind of discovering the molecule of heredity, as well as the structure of the molecule of heredity, DNA and RNA. I've given you the briefest glimpse into the central dogma, which is this idea that you read DNA, you make RNA, and then from that RNA you make protein, and then protein leads to the traits and behaviors and characteristics that we see in people. And we're about to jump into talking about DNA replication, how DNA is copied. And this is important because you have the same DNA in every cell in your body. Pretty amazing that it can be copied with such high fidelity. Well, so how does it all start? You start, as much as you might not want to think about this, as a sperm cell fusing with an egg cell. Both of these sperm cells, or this sperm cell, has 23 chromosomes, and this egg cell also has 23 chromosomes. And that combination leads to 46 unique chromosomes all coming together to form you as a zygote. And then you grow and divide. So this fertilized egg grows and divides. Well, first actually it just divides a lot before growing. But eventually it's going to grow. But it divides into many cells. This is called a blastocyst stage, when it's a hollow balls of cells. And every cell in here is totipotent. And what totipotent means is that every cell in this mass of cells could become any type of cell. It could become a neuron. This could become a stomach lining cell. This cell could become, this cell could become part of your toe. This could be a muscle cell. You get the point. All of these cells are totipotent. They could become anything. And the reason why they can become anything is because they all share the same genetic information. This cell has 46 chromosomes. It's the same as this original 46. This cell has 46 chromosomes. Again, the same as the original 46. This cell over here has 46 chromosomes. You get the picture. You're going to be learning about this process, how 46 chromosomes are duplicated and then divided into two new cells. Well, you're going to learn about the DNA replication part now, and then mitosis, how it's divided into two new cells later. But it's important for you to know that every cell in your body has the same genetic information. That's actually not quite true, but for now, believe it is. Some of the cells in your immune system that make your antibodies actually have slightly different DNA. But that's an exception to the rule. The rule is every cell has the same DNA. Learn that rule. Okay, so these cells are all totipotent. They can become anything, but over time, they specialize. How do you spell specialize? Oh my God. This is how bad it is, guys. Okay, they become specialized. S-P-E-C-I-A-L. I can't screencast and spell at the same time. That doesn't even look right. I guess it is though. Okay. They become specialized, all the cells in your body. And this process of becoming specialized is called cellular differentiation. And even though if you were to look at, for example, a neuron in your brain, you would still see the same 46 chromosome as a stomach lining cell. This is a very thin person, as well as a muscle cell in your body, or in your calf here. But the reason why these different tissue types look different, have different functions, and perform different actions is because they each take part of the DNA and they read a different section of it, and they make different proteins from different sections. So cellular differentiation is the process of a cell turning off some genes in the body and turning on others. So even though every single cell has the same genetic information, they express different parts of that. Now, you can take any cell. We now have the capacity to do this. You could take any cell in your body and you can de-differentiate it. You can unspecialize it and you can return it back to a kind of totipotent state. And then you could stick it in an egg cell. You could take the DNA out of that egg cell, stick the genetic information that you took out of someone's, let's say, calf cell, and you could now have 46 chromosomes in this egg, and you could clone that person. Okay, but we're getting a little off track here. So you have the same genetic information in every cell in your body, even though they look very different. And the process by which they become specialized is differentiation, and that has to do with turning on and off certain genes. We're going to talk about gene control and how that's accomplished later. Okay, but every single cell in your body, let's say you're to take it out. You were to take out, a neuron, and then a stomach lining cell and a calf muscle, and you took the contents of the nucleus, and you spread them out and analyzed them, you would create what's called a karyotype. A karyotype is an analysis of the chromosomes within a cell. And so a karyotype, no matter what cell you take it from, is going to look the same in terms of you're going to have the same 46 chromosomes in that karyotype. Now, the way you organize a karyotype is you start with the largest set of chromosomes and you line it up until you get to the smallest. And then these two sets of chromosomes here, are slightly special. These are called the sex chromosomes, and they determine whether or not someone will be, male or female in terms of their sex, not necessarily their gender. Okay, so if you have two X chromosomes, then you are female, and if you have an XY, then your sex is male. Not all people who are XY, for example, even present phenotypically as male. And some people who are X, you can have a whole array of different secondary sex characteristics that is genetically encoded depending on the types of chromosomes that you have in this set here. But more on that later. This person is female. The sex of this person is female because they have two X chromosomes. Okay. Anyway, moving on. So, this is the largest pair of chromosomes, and this is the smallest pair of chromosomes here in terms of the length. Now, if we were to go through this karyotype, say this is your karyotype, and we were to identify every chromosome, we could go through and say, oh, you got this chromosome from your biological father, and you got this chromosome from your biological mother, and then you got this chromosome from your biological mother, and you got this chromosome from your biological father, and so on and so forth. You can go through this whole karyotype and determine which chromosome came from which parent. This is because in your original sperm and egg, your biological father delivered 23 chromosomes, 1, 2, 3, 4, 5, so you get the point. And your mother also delivered 23 chromosomes to you, 1, 2, 3, 4, so on and so forth. So your karyotype is made up of chromosomes, one from your biological father, one from your biological mother. Okay, and you are unique. Yay, because this is a unique combination of genetic information. You're not the same as your mom, you're not the same as your dad, you are 50% of each of them. Okay, what else do I want to talk about? This. So, a chromosome, as we mentioned briefly in class when we did the strawberry DNA extraction, is a combination of genetic information in the form of DNA as well as protein. But the protein is serves a structural purpose rather than a genetic heritable purpose. What I mean by that is that the DNA strand, which you know and love, is this double helix where we have C and G and A and T bonded together, maybe hundreds of thousands, millions of base pairs long, actually, long strand of DNA. And this strand of DNA has to be organized and compacted because you actually have 6 feet of DNA in every single cell in your body. It's either 6 or 9. I've got to look that up. Anyway, there's a lot of DNA in every single cell in your body. If you were to unwind it all, it would be very, very long. So in order to organize the DNA, it has to be packaged. So this DNA is wrapped up around proteins called histones, and the histones, the bonding between the histones themselves and the DNA in the histones is accomplished through hydrogen bonding. This is why salt can disrupt this process and cause the histones to fall apart and the DNA to fall off of the histones. So it's coiled around the histones into a structure called the nucleosome. The nucleosome is a combination of histones and then DNA wrapped around it. This itself is coiled, and then you get supercoils of those coils, and then that's all also coiled up into form a chromosome. This X-like structure of the chromosome it's a duplicated chromosome. We'll talk more about what that means later, but in brief, this is a chromosome. This classic X that you see here is a chromosome after DNA replication. So this side and this side are genetic clones of one another. Okay, in this image, we have unduplicated chromosomes. So you have one piece of DNA from your dad and one piece of DNA from your mom. After DNA replication, it would look like this. You'd have an X from your dad and an X from your mom. Each side would be the same. Okay. But we're kind of getting ahead of ourselves here. So, what else did I want to say? Oh, yes, I wanted to talk about genes versus alleles. Okay, so if you were to look at a human karyotype, you would actually see that, and then you compared it to another human karyotype, you would see this, that these two karyotypes have all the same genes. I've said this in class before, but a gene is a location on a chromosome. And because we are all humans, we all share the same genes. So it's a location on a chromosome that encodes a protein. So everyone watching the screencast has the same gene. We all have different alleles. And alleles is what makes us different from one another. It's the sequence of bases so A, T, C, and G in a gene location. Okay, so what do I mean by that? Well, imagine I'm going to pick a random, a random gene. Let's just talk about a gene for height. Although there is no one gene for height, imagine we have a gene that controls the amount of human growth hormone that's expressed in a person, and it's found on chromosome 3. We would all have the same gene on chromosome 3, this protein that regulates human growth hormone, but we might have different variants of that gene. So someone might have a variant, a sequence of bases that leads to this gene creating a lot of human growth hormone. And someone else might have this same gene, but a different allele of that gene that causes only very small amounts of human growth hormone to be produced. So the same gene resides in this one location. It's the gene that regulates human growth hormone, but there are all sorts of different variants that can be found at that location on the chromosome. Okay, I think that is everything that I wanted to communicate today. So, I hope you enjoyed. See you all in class tomorrow.